Hello, my name is Soledad Serrano and I will present this work titled Evaluation of Plant Consumption by Two Species of Grasshoppers in Northern Patagonia. The Patagonian steppe is an ecoregion located at the southern end of South America in Argentina, encompassing plateaus and foothills as well as wetlands. The steppe wetlands are important environments because of their environmental and ecological functions, including the provision of food resources for domestic livestock. These ecosystems are hydrological drains, which means that there is greater availability of vegetation and palatable plants for herbivore consumption. However, humid environments can suffer drought, affecting primary production. This can trigger competition between herbivores for the limited food resources. Historically, these environments have suffered degradation caused by overgrazing, resulting in changes in the quality, abundance, and composition of grassland plant species that can promote outbreaks of herbivorous insects. One of the most representative groups of these insects in the steppe wetlands of northern Patagonia are grasshoppers. Several species have been found that forage in wetlands, some causing considerable damage. Consequently, it is important to evaluate the effects caused by grasshoppers during outbreaks on crops and vegetation in natural areas, as well as the feeding behavior that they present. In order to evaluate the diet of two grasshopper species from this region, their host plant consumption was compared using the two species included in the diet. The study was carried out in northern Patagonia in Rio Negro province, Argentina. Both grasshoppers, Dicropus elongatus and Dicropus vitigerum, are native species. D. elongatus is found in natural pastures and in different crops in South America and is considered a pest of economic importance. D. vitigerum is distributed across northern Patagonia, feeding on both grasslands and crops. Different studies of their diet show that these polyphagous species consume a range of graminoid and herbaceous plants. For this study, the two most abundant species known to be included in the diet in the field were used, Juncus balticus and Trifolium repens. Individuals and host plants were collected during the summer season of 2018. The grasshoppers were collected using an entomological net and placed in a wooden cage with a mosquito net. They were transported to the laboratory where they were maintained under semi-controlled conditions. To evaluate feeding behavior, a series of no-choice experiments were performed. After 48 hours, one day for acclimatization and another for fasting, individuals were placed in a plastic container and offered a food ration of the plants, Juncus and Trifolium, for 24 hours. The amount of plant consumed by individuals, the response variable, was obtained using the difference in plant weight from the beginning and the end of the 24-hour period. A total of 63 adults of D. elongatus and 74 of D. vitigerum were captured and taken to the laboratory. Results of the no-choice experiments were analyzed using a Mann-Whitney U-test as the data were non-parametric. A significant difference was observed in the plant consumption of D. vitigerum, with a higher consumption of Trifolium compared to Juncus, whereas no significant difference was observed in the consumption of D. elongatus. In conclusion, we observed that both species D. elongatus and D. vitigerum present different feeding behavior preferences under semi-controlled conditions. D. vitigerum displayed a higher consumption of Trifolium than Juncus. These results contribute to the knowledge of the feeding habits that these native insects present. Complementary study of the factors that define this behavior are required in order to determine the feeding niche that they occupy. The study of the diet of native insects that inhabit steppe wetlands in northern Patagonia provides useful information for pest control programs and contributes to the conservation and preservation of these valuable environments. Thank you for your attention.